Nowadays, I work mainly with biotech companies and companies that are doing some sort of a pioneering medicine or vaccine or biotherapy. So trying to do something for the first time. And there are a number of places where they need to use statistics to answer questions. Pretty much every week, I get a question about comparability. In the pharmaceutical industry, that means you've developed a process and you've made some material that's gone into clinical studies. And if that clinical study was successful, then you probably need to transfer the process or make improvements to it. And so then you need to demonstrate comparability of the material made at larger scale or a new site or with improved methods, that it's comparable to the original clinical material. So I get these questions, very regularly, and people will supply me some data to look at, but what's always missing is what is the question you're trying to answer? Comparability by definition is comparing one thing to another, and the essence of it for pharmaceutical comparability is comparing the proposed commercial material to the clinical material. But people just forget about that part and they gather data and then say, okay, is this comparable? Have we demonstrated comparability? And I'm always going back to that question of what are we trying to compare to what? What's your before state and what's your after state? What changed between the two? Which set is the reference set that represents the clinical condition? One of my favorite projects ever was working on a vaccine. It was a viral vaccine, so it's a very complex process. There are many steps. It takes about 9 weeks to get from the beginning of a batch to the final test result. And that's best case. It can take even longer. So there were many variables measured all along the way. And there was a time I was leading a team that and our goal was to figure out why we had an increase in yield. And of course, in most industries, an increase in yield is celebrated, but in the regulated pharmaceutical industries, an increase in yield could be a good thing, but only if you can explain why it happened, which should make all of us feel better that we're required to understand the process and to be able to explain results. So we were working on this problem, and we had a lot of data. We put a lot of effort into gathering data. Much of the data had been on paper, so we gathered it all and put it into digital form. It's a lot of work involved. And then we organized it by process step and by time, and we organized it so we could analyze it. And then we used a lot of sophisticated multivariate and univariate methods, all sorts of things, principal components analysis, bootstrap forest, many methods. And we did find very strong clues to what had happened to cause this yield increase. But the problem with data like that we call happenstance data. It's the data that has been generated naturally as we run the process. So there was no intention to push the boundaries of the process controls. It was just data that naturally occurred and was collected. So we could get clues to what happened, but data like that can never be completely definitive about cause and effect in the process, because by definition, when we run that process, we're attempting to control it within narrow ranges. So there may be impacts. For example, temperature is a simple one. So temperature might impact the yield of the process, but because we're always trying to control temperature to a narrow range, we may not be able to see the impact of temperature from this happenstance data that is just collected as the process runs. We exhausted every method we could dream up to understand what was in the data we already had, because data is precious. We never want to ignore data we already have and find everything we can from it. But then there was another stage where we needed to explore real cause and effect. and to push the boundaries. I do remember George Box would say, if you want to understand the impact of a factor on a process, you need to intentionally change that factor. You can't just wait for it to wander around and see what happens. You need to go in and change things. And that's where design of experiments comes in. I think that's the gap between artificial intelligence, machine learning, all the big data methods that rely on data we already have in hand, versus design of experiments, where we say we've perhaps operated a process in a fairly narrow window by design, but we want to understand, maybe there's an even better yield that we could achieve, reduce the cost of the product, but because we've not explored factors like temperature beyond their normal operating ranges, we don't even know it. It's invisible to us. And the big data, machine learning, artificial intelligence may miss that opportunity outside of the existing data region. Where design of experiments, we can say, the data we have gives us a clue that there may be an impact of temperature. We don't know how big it might be, but we see this suggestion in the data that it could be important. So what experiment should we design? Maybe we have a lab scale version of the process, and we can go run it and intentionally push temperature beyond the region we normally operate, along with other factors in combination. In a designed experiment, there may be some sweet spot or some combination of temperature and cell density and the quality of the feeds to the process, that if we can dial all of those in to some sweet spot operating level, we may double yield. I mean, actually, I've seen that where and even better results sometimes, because we get kind of in a bubble of, well, we've always run it this way, we know it works, don't change anything. But we may be running along for years sometimes, missing an opportunity just beyond that that could have really great impact.